I want to start off, I guess, giving you a little background on this. First of all, thank you for all being here. I'm really honored to be able to give you guys a little bit about what we've been doing for the last few years with the Aquarium and with a bunch of other partners. This project, it's hard to call it a project, if you will, but this really started with a long-term conversation between about 5 people. Charmaine, Adam Frederick of Maryland Sea Grant, Setso Bachvarov, and David Flores of Blue Water Baltimore. David Flores was the waterkeeper for Blue Water Baltimore. And we would talk to each other about the questions we'd get about the Baltimore Harbor. People would ask us, because of where we were and what our titles were, they'd say, why is the water this color? And what's going on with that? And why do I see a dead fish? And aren't there a lot of crabs today? And we really struggled with how to engage people and explain in a way that doesn't take a lot of expertise and a lot of specialized knowledge to understand what's happening. And that's hard, I think, because a lot of what happens in the harbor and a lot of what's driving what happens in the harbor is very policy-oriented. It has to do with sediment and nitrogen and phosphorus and dissolved oxygen and water quality and chlorophyll. And I think that for most of us, those don't mean a lot. You hear them and you think you know what they mean, but it's not like you can put your hands on it and talk about it. And so we were really struggling for ways to explain the phenomena and the biology. And I think that what Charmaine and I are going to tell you tonight is it's something where we think we've got kind of getting there. So I'm really appreciative to those people who had that conversation. And it took a long time to develop the ideas and also to gather the resources. So at the end, we're going to talk to you about how you can get involved. And I didn't put on my list, but I'll say now, one of the ways you can get involved is to support science funding for everything, but really because the decisions that we make in society really depend on good data. So with that, I'm going to introduce one of the other characters in this talk, sort of leading it off, and that's a creature you're probably all familiar with. This thing right here. We filmed this creature just a few yards from here, and they show up every year. This is Karlodinium sapidus, the blue crab. Does anybody know what sex this animal is? Yeah. Jack. Pink fingernails, Jack. This is a female crab, so the female, you can tell it's a female because she has pink claws. I'm showing you this because this crab is telling us something, and then it was sort of an aha moment when I realized that you can tell the story of the harbor just by focusing on one animal and carry that narrative all the way through. So this animal comes to the harbor not by accident. In fact, she had to travel about 200 miles to get here. She started her life in the Atlantic Ocean as a microscopic larval crab that was released there by a crab in the lower bay, spent a month in the coastal zone, resettled into the estuary, and spent probably between 3 and 12 months making her way back up to Baltimore. So she didn't get here by accident, and she had to spend a lot of energy to get here. That tells us that the harbor has an ecology. She has reasons to be here. But we're going to tell you about that, and we're going to tell you about some of the stresses in that. I'm not going to tell you that. The first part of the story, I'm going to hand it off to Charmaine, who's been a fantastic partner in this, and the Aquarium has been so supportive in all of these studies. So I'll give it to Charmaine. I missed my next cue, which is I'm going to tell you what we're going to tell you, so in case we don't get there, our cue is we're going to tell you about the history, why we need to know, and the data gap, the information gap that we have right now. We're going to talk a little bit about this project that we're doing and all the people that are engaged with that and how you can get involved with that. Thanks.
Thank you. So hello, everyone. Thank you all for being here today. I was talking to some people outside. I know that we have people from the community here as well, so that means a lot to me. So thank you all for coming. If you can't hear me, though, in the back, you can do one of these little things, and I'll speak up. So I just want to make sure everyone can hear me through this. So like Eric mentioned, we're going to talk about the history of the Inner Harbor. And to understand the environmental condition that it's in, we have to know where we're coming from. So this is a photo from 1792 of the harbor. What you're looking at here, we have the Jones Falls that comes down. At the base, there's a wetland. To the right of that is Hartford River. But if we looked at this and we overlaid the current shoreline of the inner harbor, this is what we would see. So now what you're looking at is all of this light blue or this aqua here, that's actually land now. So we've filled in the harbor. The Jones Falls now becomes I-83. The Hartford River is now gone, so it is completely developed over. And all of this, like I said, are these industrial piers. So how did we get to that shoreline? This is a photo from 1930. This is what the harbor looked like. And at this point, we have completely dredged all of our shallow water habitat. So it is no longer there. The harbor is now much deeper than it was. But despite the negative environmental impact it had back then, Baltimore is flourishing. So Baltimore at that time is a transportation hub. So it's connected to the rest of the world through its industry. We also reached our peak population. So in the 20th century, our population is somewhere around, it peaks around 900,000 people. Just for comparison, today, the population of Baltimore City is around 600,000. But what does this do to our economy? I want to point out that the success of the economy in Baltimore City at this point is driven by its Baltimore-based industries. So I want to provide an example of what that looks like and what that means. So this is a photo on the left from 1979, and then on the right it's 2016. This is Harbor East, just around the corner from here. This is the former location of the Baltimore Chrome Works, which opened up in the 1800s. It was later purchased by Allied Chemical in the 1950s, and then later purchased by Honeywell in the 1980s. And it's a chrome site. So what they did was they would import raw chromium ore. They would extract the chromium and then use it for production. So they would be used in mostly for the steel industry, but also for leather tanning and for chemicals like paints and things like that. What's important to note is that the activities that took place here are most of them happened before the establishment of the Environmental Protection Agency. And all of the activities occurred prior to the establishment of the Maryland Department of Environment. I have to be careful what I say because I have an MDE employee in here. But there is waste generated. So waste generated over 130 years by the activities of this plant. And that waste was used to fill in portions throughout the inner harbor. So to create more land. And they did it at that site as well. So they wanted to increase their footprint, to expand their operations. So it wasn't until the EPA kind of stepped in just after the 1970s, after they were established, in order to clean up of this site, which led to the 1989 consent decree. And what we know now is that this site remained abandoned for many, many years until it was capped and then developed over. So let's jump into the 80s. August 8th, 1981. This is a very important day for the Inner Harbor. This is the first day that the National Aquarium opened up its doors. So this is when we opened. And that's a picture of opening day. And at that time, we have a changing approach to the harbor. So as industry dies off, there's a gap to fill. And that gap they want to fill with tourism. So now they want to make Baltimore a tourist destination to bring people in. But we're still really not worried about the harbor's condition at this point. But now, and we look at where we are today in 2019, again, we're faced with that same changing approach to the harbor. But this time, we're focusing on the harbor itself. So the harbor has a different purpose. And the harbor wants to be used. We have people out there that are fishing. We have people out there that are boating and that are kayaking. They're using the harbor for recreation. And even though the heavy industry is gone, the system is still stressed. So we see the trash out there. All of us see the trash out there. But it's also the polluted stormwater runoff, and it's the sewer sanitary overflows that feed this excess nutrients into the harbor and lead to these incredibly intense algal blooms, which are causing damage. So I want to spend just a few minutes to talk about what's occurring in the harbor. This is the most common question I get when I'm out working in the harbor. Why is the harbor colored that way, and what does it mean? So I'm going to touch on the three major things that are most likely happening in the harbor. Starting with our mahogany tide. So that's what this one is over here. This is produced by a chlorophyll-rich dinoflagellate called Prorocentrum minimum. It starts in the spring, and it'll pretty much last the entire summer in different degrees. But when we do get these really high spikes of this algal bloom, we have a massive die-off. And then that die-off depletes oxygen from the water. And then what that leads to is a fish kill. This next one over here is actually really quite unique. So this is the sulfur bacteria bloom. This one is, you're going to see this one in the fall. So what happens here is there's a change in temperature from day and night that's very drastic. So we have, in the fall, if you remember, you might have a really hot day followed by a really cold night. What that does is drastically change the temperature in the harbor, and it causes a change in the density. So the surface of the water actually sinks to the bottom because it's heavier and it's colder. The bottom of the harbor is then displaced and moves to the surface. And it's going to bring up with it the sulfur metabolizing bacteria. So that actually reacts to the sun. It's really colorful, and you get this milky green color, or what we call our pistachio tide. This one, again, because there's no oxygen at the bottom of the harbor, what it does is it leads to another fish kill. So then our last one here, this one is actually unique in that it is studied heavily right here in imet. So this is our Karlodinium beneficum bloom, another chlorophyll-rich dinoflagellate. This is a little bit harder to see. This photo is taken from the World Trade Center, looking down at the National Aquarium, which is here. It does have a little bit of a red tint, but it's not that drastic, so you can kind of see it over here. This one is studied because it releases carlotoxins, and those toxins get into the gills of fish and make it very porous. So it's directly linked to killing fish. So these are all bad. Not that great, but what's important to know here is that we have organizations that are advocating to clean up the harbor. And this is just an example of just a few of those. The Waterfront Partnership, who is represented here today, is a business-oriented nonprofit. And then we have Blue Water Baltimore, which is an advocacy group. And what's great is that all of what they're doing is supported and backed by the Clean Water Act of 1972. All right, so where do we stand today? Let's look at where we are. This is the latest report card that was released by the University of Maryland Center of Environmental Science. This is 2018. And this is the entire Chesapeake Bay that's graded. We're in the Patapsco, and we're in F. Not a big surprise. We have to know what that F is, though. So the issue with the F is that it's related to the low oxygen levels in the inner harbor and also the algal blooms. And the result of that is a low biodiversity. So theoretically, we know that. But we don't have the science to back us up. I use this a lot in a lot of my presentation because I think it speaks volumes. But this is an open source GIS-based map that you can get from the Watershed Resource Registry. And I turn on a bunch of layers that are related to fish populations, surveys that we know, anything that has to do with wildlife. And we have all this amazing data all throughout the state of Maryland. But if you look at our cities, Baltimore City and even Washington, D.C., there's this gap of information. So this is where we went into trouble. But looking back at that F and knowing that lowering nutrient loads could take decades to do, we have to ask ourselves, what are we going to do today? So for the National Aquarium, this is our idea. This is a floating wetland, and we want to build these to address the issue of a lack of habitat and a lack of structure in the inner harbor. So this is a rendering of what we wanted to build, and then this is our vision of what we hope to accomplish. So this is on a larger scale, bringing back some of that lost shallow water habitat that we were missing from back in the 1930s when the harbor was dredged. But we're not only with this project providing the habitat, but we're also connecting the community because people want to use the harbor. So this gives them the access to enjoy a natural space that brings back the wildlife. But before we invest in a capital project that is very expensive, we have to know how to measure its success. So how do we do that? Well, we have been, and we're going to do that through prototyping and then using a bunch of methods to measure the biodiversity. And that's what I want to focus on the rest of this presentation with. So here's our prototype. This is August of 2017 when we first installed it. There's a lot of floating wetlands out there. They are not all created equal. This one was custom built, so we have different topography levels. We have this nice channel, this open channel that runs down the center. It sits on a fiber grate platform, and it can ride up and down the tide with those pilings there. But it also is completely out of the water because we have these pontoons underneath that can control where it sits in the water column. So we have complete control over that. So as we're planting that, we want it completely out of the water to make it a lot more easier. This is what it looks like today. It's out there, so you can take a look. And you can't see it, but that center channel is still there. When we take water quality readings, we've had some pretty extreme highs of blooms and temperature. So this past summer, we were taking readings of the temperature of the water of the harbor in the 90s, in the mid-90s. It is a very difficult environment for a fish to live in. That center channel is actually cooler because it's pulling water up from underneath the island, and it's shaded under there. So it's a safe haven for a lot of these fish that are living out there. So we know this, but is it working? So we did a little experiment. The island is out of the water. That's myself and Jack Cover, who's over here. But we slowly sank the island, and this is just after we launched it, so probably within that week or a week after. And we wanted to know what would be coming into that channel as we slowly sunk the island. These are photos taken about every 3 seconds. And if you forgot where you were, you can check this guy out coming by. It's an Utz bag. But the second that channel becomes submerged, what you're seeing is this flood of fish kind of moving in. So these are Atlantic silver sides. They're native. They were most likely living in the harbor already, but didn't really have a refuge. So they immediately take refuge, and there's hundreds of them. There's one mummy chug there, too. He's a little lost. Okay, so we do the same thing a year from now. So now what we want to look at is, does it change, or is there any diversity? So now our island's a little bit more mature. It looks a little aged. The grasses look a little bit more mature. And we have the same thing, but now we're having a little bit of more diversity. So we have some killifish, a banded killifish. We have mummy chug. But even more surprising, we have this little guy. So that is a common snapping turtle, native to this area. And this is something that we would never have been able to capture unless we had that GoPro there. So we are testing other habitats in the harbor. So this is another prototype, and I want to give you a place of where this is. So if you're out in the harbor, this connects. There's a pedestrian bridge that connects Pier 3 to Pier 4, and it kind of bounces when you walk across it. Everyone likes to jump. At the base of that, it's held up by these big pilings, which happen to be the perfect depth in water for a horizontal oyster reef. So this is Jack and I, and we collected oyster shells from the restaurants nearby. So they, instead of being discarded, we use them. So they're not live oysters, these are just shell providing habitat. But will it provide habitat? So again, we go back to our trusty GoPro. This over here is a male pumpkin seed sunfish. It gets that name, pumpkin seed, because of that little orange spot on its side. And this is a very common mating behavior. So this male is protecting this nest and keeping all the soot and any sediment off of the nest so it can survive. So in addition to those types of habitats, we also have vertical oyster reefs that we call biohuts out in the harbor. So this was adopted from the French. They did this in the Mediterranean. But the harbor is very different. So this is just some footage near the floating island. And again, we see our pumpkin seed sunfish, except these are the juvenile ones. So these are the very young fish. And they're kind of hanging out by the biohut because it's a safe haven for them. So they're interested in the camera, but they know if anything came after them, they could dart back into their biohut and be safe. But this biohut is serving the purpose of a habitat, but also as a food source. There's oyster shells in one half, and then it's empty in the other. And you can see there's a tremendous amount of what we call our glass shrimp or our common grass shrimp. And then even growing on the biohut, if you look close, we have a lot of barnacles and dark false mussels. So this is great. What's even better about the biohut is that we can actually pull them out of the water and, again, bring the community into this project. So when we pull one of these out, what we want to do is inventory every living thing that's inside, because I can't capture everything on camera. And people are amazed when they see an animal and we tell them that it's from the harbor. It's an absolute wow moment for them. But we also use students as well. So this is Langston and Shima. At the time they were college students. This is Alexandria. She was a high school student. Langston has, even though he was an intern, he has since been hired full-time with the National Aquarium. I don't want to embarrass him, but he's right up front. But by using those methods and involving the public, we're able to do a lot of outreach and get people excited about the harbor again. But with this project, we've been able to either photo document or video document 34 different species of wildlife utilizing the harbor as a home. So everything that we looked at up until now are things that we can look at, that we can touch, that we can identify, and then that we can count. So it's a process that is very natural to us, and it's a way that we collect the biodiversity. But there's a reason that these animals are here. In this case, these habitats that we see, these biohuts, those oyster reefs, they become part of the food chain because they're providing the structure for biofilms to grow. So these animals are here because they're eating the food that's provided to them on these structures. So now the question becomes, how do we quantify this type of biodiversity? And lucky for us, there is a system in place. So this is a website. It's from Maryland Sea Grant. It was introduced to us by Adam Frederick, who is also here, so if you have questions, you can talk to him at the end. It's an educational tool built to count the biodiversity of those sessile organisms. So it provides the identification key and then the methods to calculate all those statistics. So this is how we implement this. We have a PVC pipe that has 5 acrylic discs on it that are very clean and brand new. We deploy that into the harbor. It sits about a meter just below the harbor surface, and we just let it go. We allow things to grow on it. So this is what it begins to look like months later. So slowly there's a accumulation of this biomass. So all of this is biomass. So this is us just sampling. We sample once a month. So this is Adam and I sampling, but also, again, we include our community. So these are two high school teachers who have brought busloads of students down to explore the harbor, to look at these biodiscs. And these teachers spent the summer with us to learn the process and the methods so that they can go back to their classroom and implement that in their classroom. So this is just a series of one sampling season. So in April, there's not a whole lot of growth. This is our peak. And then in December, as the temperature starts to drop, we have a little bit of a die-off. So there is a die-off between here and here. And then what I also want to point out is you'll notice on this particular disc, there's a little bit missing. And Eric is going to get to this later in his talk to talk about the DNA processing that goes on with that. So each month, we take them out to sample them, but we also take a scraping for DNA analysis. So just looking at biomass, just the growth of what's on there. We have two sites that we're looking at. So our floating island is by the east site, and this is our comparison site, the west. This is from this year. This is just weighing the discs, so how much biomass. So clearly September there was a big peak. In October everything died off. So something happened between September and October that we have to investigate. Looking at our comparison site, it's different. So we get a peak in October when everything dies off in October from our other site. So it's telling us something, but we're just not sure what it's telling us just yet. So let's dig a little bit deeper. When we take these discs to sample, moving beyond the biomass, looking at it and weighing it, we're going to continue using these methods, and we take these discs and we put them under a microscope. So we bring them into this building right here, up into a lab, and then we blow them up on a screen and we start to see what's living there. So this is a video. So these are two amphipods arguing. So this is a May sample. So the disc has only been in there for a month. So there's not a whole lot going on. This community is just getting kicked off. But yeah, those are the amphipods. We also have, So we also have some, let's see, the hydroids are here. And then we have colonial ciliate that you see in these, which are a little hard to see. The hydroids are closely related to jellies. They're stinging organisms. So looking at May, now we're going to move to September, and now we have some diversity to look at. All right, so in this one, now that's more mature, we have that active barnacle that's down here eating. And we also have, what you're looking at are dark false mussels. So the dark false mussels, with these arrows here, point to the siphons that are actively pulling in harbor water to clean it. And then we have some tube bryozoans up here. So it's a much more dynamic system, but with this method, we can now look at these organisms, we can identify them, and we can count them. And this is what gives us our biodiversity. So this is just a snapshot from 2016. This is the entire sampling season in looking at all the animals that we were able to count. And when you look at this, I call this muck because that's what it looks like to me. It looks dirty, it looks slimy, but what you need to know is that every one of those organisms is cleaning our water. So even though it doesn't look good, it's doing a lot of work out there to help improve water quality. So in 2016, we had a great year for diversity, but I want to point out the dark false mussel. So the number here is low, and in 2016, we didn't have a tremendous amount of rain, so that kept the salinity pretty high in the harbor and kept the dark false mussels at bay. So they kind of behaved themselves. What we saw in 2018, no surprise, but as we know, 2018 was the wettest year ever on record. And that opened the window, that fresh water opened the window to have these dark false mussels just absolutely explode. And you can see our acrylic discs are just completely taken over. And what this does is drastically lower our biodiversity in the harbor. And they pretty much will grow on any hard surface that's under the water. I'm not sure how many people scootered here, but I would appreciate it if you kept them above water. But yeah, they will attach to any hard surface. All right, so at this point, I'm going to pass it off to Eric.
So, all right, at this point I'll pick it up and talk a little bit more about mussels and deeper than that. So the mussels have really engaged people's imagination. This is a picture of high school students from the park school last summer. They'd heard about our project through a network of people, and they wanted to come down and see what we were doing. What you see here are some students looking at a baby crab. If you haven't held a white-fingered mud crab in your hands, you're missing something. They're very cute. You're going to want to rescue it and put it back in the harbor. But these students are watching, actually, the K through 12 education people here at SciTech. We've got a couple of them here in the audience, pulling discs out of the harbor that are cultured at different depths. And this is part of a program at SciTech to teach kids about the importance of oxygen in biodiversity and abundance in the harbor. And so what we have here is a natural laboratory with that depth profile. It's unfortunate, but we have a natural laboratory that deeper in the water you have less oxygen. And so these students are investigating discs that were taken out from different depths, and they're going through the same process that we just described to you. And then they report back and they think about those concepts and what's going on. The National Aquarium now, with funding from NOAA, has a program to bring this kind of messaging and this kind of investigation to sixth graders. And if I'm right, I think Charmaine said that every sixth grader in Baltimore City is coming through here in a NOAA-funded program to expose them to this. And you can just see the excitement of the kids in seeing that there's life in the harbor. So I think the opportunity here for exposing every sixth grader in the school system to this setting is, I think it could be transformative. So this is really wonderful. So we've talked to you about the overall picture. Here's that water quality issue. We talked about big organisms that you can see. We talked about that food web and what's on those sessile organisms that are on those structures. But as Russell said in his introduction, here at imet, we're experts at digging into the genetics, the underlying genetics of what's going on with things. And so our expertise is to add and complement these by looking at the DNA content of these communities. So we're using a method that's called DNA barcoding. I use that so much I forget to explain to people what it means. In the same way that a barcode on a product is kind of an anonymous set of lines and numbers that can tell you what the product is if you have a reference database somewhere. DNA barcoding is very much the same thing. We can go into the DNA of any species, find a particular gene whose sequence is characteristic for that species only, and then if we have the right database, we can just get DNA sequence and then know what species we have. So we're using that method in the harbor system. So this disc that Charmaine showed you before, with a little bit of the muck scraped off, that muck got put into a freezer, and then at a convenient time, a student or a volunteer with a nonprofit or somebody else in the lab extracted DNA from it, and they took that community, got all the DNA out of everything at once. So there's that whole scrape, the DNA goes into one tube, and then we can sequence. We have methods to sequence what we call, the barcode gene from everything in that sample at one time. So that creates this great mixture of sequences, but we can then, using a lot of computing power, we can take those and figure out what's in there. So here's an example of some barcode sequence at the top, and schematically I've now represented that community as the bits of DNA, right? The green, the blue, and the red, the squares, the star, and the circles. We take these many, many sequences, compare them to global DNA databases. Right now, as of today when I checked, between GenBank and the Barcode of Life database, there are about 9 million barcode sequences in global databases that are accessible to us. So there's a very good chance that any DNA sequence we find, someone has found and authenticated in the database. And in this hypothetical example, let's say that the green sequences might have been bryozoan A. It was maybe the more abundant species in that set. And that the orange-red sequence was, say, a bit of a clamworm. So this is sort of how it goes. All the work that's happened so far has happened either through summer interns or students, or one of the best contributors to this project has been the Baltimore Underground Science Space Group. These guys are I should let them explain it, but they're a nonprofit that really strives to bring science to everybody. They're really trying to take the experts out of it and say, this is accessible to all of you. So many people walk up to us and say, what you do is complicated and hard and impossible for me to understand. And it actually isn't. And Bugs is proving that. So here's Youngbi and Syra and one of the founders of Bugs, Tom, working in the lab. This is an action shot from a few weeks ago. And what they're doing is they're amplifying those barcode genes from the DNA samples that we have, and they're running gels, and they're going to process that. I'll explain that. And here they are at the imet open house this spring, showing the public what they're doing. And also in the audience today is a UMBC artist who's joined us. Stephen Bradley is a member of Bugs and has worked on the DNA sequencing side, but he's also got some art projects that he's done with the material that he's getting from us. So I'll show you that as well. So here you go. This is about as geeky as it's going to get, so don't get too scared. This is what the guys at Bugs are seeing. They're amplifying the DNA, the barcode DNA. This little band here is the amplified products of that reaction. And within a single band, you may have 10,000 or 100,000 copies of all those genes that represent all the species that you extracted the DNA from. So the output sequence might look something like this, except you'd have a data file that would have maybe 10,000 or 100,000 copies of this. They'd all be different sequences. Many of them would be the same, but many of them would be different. And you need computers and computational power to figure out what's what. So what we do is we'll take those sequences. I say we. Really what we're going to do, Setso Bashvarov, our bioinformatician and evolutionary biologist, takes these sequences and basically clumps them into groups. So let's say that out of those 374,000 sequences we got in 2016, he's found that there are 143 types of sequences that are represented more than 100 times in this group. And you can graph that and just kind of say, all right, we get an idea. There were a lot of these sequences and not very many of these sequences. So given what we've seen so far with the mussels, you can imagine on a year where there was an explosion of mussels, a lot of the DNA are going to be the dark false mussel. If we look at 2016, the most abundant DNA was actually that of barnacles in July. So this is a graphical representation of the sequence analysis that Setso gave us. This big arc here is barnacles. We've got another big group here of marine worms. So this is one of those mudworms that you saw waving its arms around in the video that Charmaine showed you. Down here, the dark false mussel, only 3% of the sequences. And then we have some bacteria, a number of protozoans in this group called the alveolata, and then the green wedge, 19% sequences that didn't have a match in the database. So these aren't necessarily undescribed to science. They just that no one's ever gone out there and taken a single organism, sequenced it, and put it in the database. Okay, so that's July of that year. And then in December, we can compare what happened over time. We can see that by December, the barnacles now are only a small percentage of our sequences. And what's replaced them is a whole lot of copepod DNA. Copepods are tiny crustaceans that live in the water, but they can also grab onto things. They can also be eaten by things. So there's some questions as to why we have so much copepod DNA in here. It could be that they're hanging onto the disk, or that they were eaten by things in the disk. You see that we have now the protozoan group that was so small in July is now really expanded, and we've got a lot of things. We've got ciliates, dinoflagellates. The Setso here, I don't know what this is. But it's a really diverse group of organisms, but they're all unicellular, and they're all less than 1 millimeter long. So it's no wonder we didn't see them in our videos, either up close or in the harbor. But now you can see how we're getting into that other level of specificity. And again, we have a big chunk of sequences that didn't show up in databases. So this is this is how we're finding new things and kind of diving down and getting a complementary set of information. I just want to point out one of the sequences we found in that early disc, that that you saw scraped off, had a whole lot of something we never thought we'd see in the harbor, which is an oomycete. Oomycetes look like fungi, but they're actually a completely different taxon all by themselves. They're associated with wet or moist environments, often as pathogens of plants or other things. And so again, we're getting a big signal for oomycetes, and I guess for us it's interesting. We didn't expect to see it, and a lot of this is about discovery. So this gives us fodder for either further investigations, forming hypotheses, all sorts of things like that. And then we can dive down even one level more, and this is what Langston and Chima did when they were in my lab over the summer, is they got interested in whether or not this little tiny worm, this is like a millimeter long worm. Mudworms are incredibly abundant out there, but they looked to see if in a single worm, how many barcode sequences would you find? What they found is that in individual worms, there are at least 6 different barcode sequences that are quite different from each other. That if we hadn't taken a single worm and sequenced the barcodes out of that, we would have thought we had 6 different species of worms. So these guys have uncovered yet, I guess, a within-species level of biodiversity. So kind of working backwards, I just got finished telling you about how we can use molecular methods, DNA barcoding, and individual sequencing to dive into the invisible or the unseen kind of biodiversity that's in those discs. And Charmaine told you about investigating the sessile organisms that are really the linchpin to the food web, and in some ways to cleaning up. I wouldn't say cleaning up the harbor, to clarifying the harbor. They're feeding on the plankton. They're taking particulates out of the water, and those are there as food for the fish, and they're changing the water quality to make a better habitat for the larger life that's there. The study that we've told you about has actually been expanded with funding from the Franz Marek Foundation. Scientists at the Center for Environmental Science down in Solomons, Laura Harrison and Ryan Woodland, are doing full-on studies of fish biodiversity, benthic grabs, oxygen, and stable isotope analysis throughout the harbor region. So here we are up in the inner harbor here, but they've gone to the Middle Branch and Bear Creek and Stony Creek and Curtis Creek. And Ryan just yesterday presented preliminary results from that study at an international meeting down in Mobile, Alabama, that's still going on. So we're engaging both with very serious science here to try to help them explain what they're seeing by looking at the biodiversity at a low level, as well as engaging the public and explaining what's happening at that level. So I mentioned Stephen Bradley's work, and I just want to show you what he's done here. I love this. I think that this is a piece from an exhibit that he had at the UMBC studio last month. And one of the things he did is he took our dozens and dozens or hundreds and hundreds of videos that we've taken for research, and he took clips of them and he assembled them into something that's completely new and gives us a completely different view of the biodiversity in the harbor. So I really appreciate Stephen doing this and opening up another window for us as scientists to be able to sort of give us permission to look at our science in a new way. So anyway, I wanted just to recap. I think that this biodiversity has scientific value, this obviously aesthetic value, and the public role in this is really important to us. It's not just that we think it's a good idea. We really need the public engaged in this. So this is the list of some of the people I talked about as our partners in this project so far. Cities and nonprofits and city schools and nonprofits and universities. And you can also get involved. Bugs would be really happy to have you get involved in either harbor biodiversity or any other kind of citizen science. And I want to hand it off to Charmaine because I think she has something that she can tell you about that's probably something you could do like right tonight.
Yes. So thinking back on that biodiversity map, there Baltimore City and Washington, D.C. were kind of this clean slate. I would like for you to look at this map. This is iNaturalist. So these are members of the community. These are non-scientists that have an app on their phone that are collecting data in their own backyards. This is a free app I encourage all of you to download it and start snapping pictures of what you see around Baltimore City. You don't have to know how to identify it, because if you take a great photo, what it's going to do is suggest what it thinks it is, and it's usually pretty close. And even if it's not close, myself and team members monitor the site constantly and are telling you, yes, you're right, and if you're right, your information goes up to research grade, which can then be used for a scientific study. But if you're wrong, then we can say, you know what, I think this is what it is. So I really encourage you to download that app and start snapping those pictures. I'll be here if you need help afterwards. But this is what the website looks like. And from that pretty much concludes what Eric and I wanted to share with you tonight. So I think we have some time to open it up for some questions.
Yeah.